Good afternoon, students. So good afternoon all of you. So welcome to Unacademy EAMCET YouTube channel, students. So myself Dr.Rajendra Prasad, biology educator on Unacademy platform. So today's class we'll be discussing about the topic. The name of the topic is enzymes. So we'll be going to discuss about the topic that is enzymes. So before going to our discussion on the enzymes, I would like to introduce about our Unacademy Plus subscription, students. So Unacademy Plus. So Unacademy Plus is for students. So Unacademy Plus is for students who Unacademy Plus is for students who subscribe to our courses, students. Unacademy Plus is for students who subscribe to our courses. So let us see what is the importance of our Unacademy Plus subscription. So let us see the benefits of taking Unacademy Plus subscription, students. So premium content. So premium content will be available at affordable price. So structured live courses and dedicated live doubting sessions. So quizzes at regular intervals, batches. So lecturers will be in batches, students. You can go to the batch which you are interested and you can join those lecturers. Then test series for daily practice papers, and all this you can get for a single subscription amount of, that is 2,250 rupees, students. So there is a 1 month trial plan of 2,250 rupees is available, students. Recently one batch course was started. Name of the batch is called as EAMCET Rankers batch. So this has been started in the month of December 23rd. Okay. So here we have well educators are there. You can see well qualified and well good educators are there. So you can join those classes by purchasing this batch course, that is EAMCET Rankers batch. And what is the importance of this batch course is, so learners for best educators for EAMCET 22 detailed explanation in Telugu language will be given, students. Complete syllabus will be covered. So tips and tricks also will be given by the educators. So this all will be going on, students. Apart from that, there is another batch that is going to start in the month of the January, from January 11th. So from January 11th, another batch course is going to start. So in this batch course, so I am taking the biology classes, students. I is taking the chemistry, and I is taking physics and mathematics. So students who are interested can join this Telugu batch. And what is the importance of this Telugu batch is complete syllabus will be covered. So tips and tricks also will be given by the educators, topic wise practice papers, and full and mock test. So this all you can get for a 1 month trial plan of 2,250 rupees, students. So use my code Rajendra Live to purchase this particular course for 1 month trial plan of 2,250 rupees, students. So that is about our Unacademy Telugu batch for EAMCET preparation, students. So apart from this, there is a long-term plans are also available. So there is a best long-term plans also available, students. You can see those long-term plans. So 6 months plan. So 6 months plan, long-term plan, 6 months plan costing around 10,125 rupees, and 12 months plan. So 12 months long-term plan costing around 12,375 rupees, students. If you are interested to join this long-term plans, use my code Rajendra Live to join the 6 months plan and as well as 12 months plan, students. These are very less amount, students, when compared to other corporate sector colleges in this AP actually, or in Telangana. So let us come back to our topic today discussion, that is on the enzymes, students. So let us discuss about the enzyme. So let us see what is this enzymes actually. If you see the chemical nature, first let us discuss about the chemical nature of enzymes. So if you see the chemical nature, so these enzymes, so these are if you see the chemical nature of these enzymes, if you see the chemical nature, so if you see the chemical nature, these are enzymes are made up of, so they are made up of proteins. So enzymes are made up of proteins, students. So especially what type of proteins they are. So we know that in proteins we have primary structure, we have secondary structure, tertiary structure, quaternary structure. Out of all those, the enzymes. So these enzymes are made up of proteins, and those proteins are in tertiary structure. So tertiary structure. So proteins which are in tertiary structure, so they exhibit this enzymatic activity. So enzymatic activity is exhibited by those proteins which are exhibited in the tertiary structure. So in tertiary structure, why the enzymes? Okay, so why the enzymes, the protein part of the enzymes which are in tertiary structure is because, so during the formation of tertiary structure, what happens is there is a formation of the pockets takes place. So formation of the pockets due to folding. So formation of pockets due to the folding of these polypeptide chains, and these pockets, due to formation of pockets, it creates an active site. So it creates an active site. So every enzyme has an active site. So that active site formation takes place. So that particular active site formation in enzyme takes place only when protein is in tertiary structure. So when protein is in primary structure, it cannot form any pockets. When protein is in secondary structure, it cannot form any pockets. But when protein is in tertiary structure, then only it can form pockets, and that pockets later develops into active site. So active site is very much important part of the enzyme. So to the substrate will bind the enzyme at particular location, that is called as an active site. So the active site formation takes place only in the proteins which are having the tertiary structure. So that is the reason. So we have to remember that. So enzymes are no doubt they are made up of proteins, and specifically what type of structure is present in the enzymes of enzymes means it is having the proteins which are exhibiting the tertiary structure. So why means reason is because they have an active site. So because of that reason it is given like that. Now let us see about these enzymes. If you see, so simple enzyme. So there is simple enzyme. So there is one term that is called simple enzyme. Means the simple enzyme means that particular enzyme. So that particular enzyme So simple enzyme. What is simple enzyme? So that particular enzyme that is made up of only protein. So that is made up of only protein. So it is made up of only protein. So that is known as simple enzyme. So simple enzyme. So what is simple enzyme is? That is enzyme that is made up of only protein. Such type of enzyme is called a simple enzyme. Then generally what is the main function of enzyme actually? Why we are using enzymes? Why we are discussing about enzymes? So what is the main function of enzymes is? So they act as enzymes. So they act as biocatalyst. So they act as biocatalyst. So that is the main function. So that is the main function of enzymes actually. So what is the main function of enzyme is? They are acting as a biocatalyst. So biological catalyst. Biocatalyst means if you see the full form, it is called as a biological catalyst. So enzymes act as a biological catalyst. So enzymes main function is biological catalyst. They are acting as a biological catalyst. So that is the main function. That is the reason. So it speed up the reaction. What is the main function of this catalyst? So catalyst generally they speeds up the reaction. So they speeds up. So this speeds up the reaction. So here similarly here also what happens is this speeds up the reaction by reducing activation energy. So by reducing activation energy. So this reduce the activation energy. So that is the main function of the enzyme. So what is the main functions of enzyme? If you see, enzymes act as a biocatalyst. So enzymes act as a biocatalyst, no doubt about it. So enzymes act as a biocatalyst. What is the main function? That speeds up the reaction and by reducing the activation energy. So that is the main function. So this is something about reduction part of enzymes, students. So if you see the chemical nature of enzymes, so the enzymes are made up of proteins. So specifically what type of structures? Okay, so proteins are having primary structure, secondary structure, tertiary structure, quaternary structure. Out of all these structures, the enzymes, so majority of enzymes are having the proteins, and these proteins are made up of tertiary structure. So but there is an exceptional case where nucleic acids. So nucleic acid is also acting as an enzyme. So there is one exceptional case. We know that majority of enzymes are made up of protein, but there is an exceptional case where nucleic acid is also acting as an enzyme. So best example is the ribozyme. So ribozyme is the enzyme. So ribozyme is the enzyme. It is made up of it is from RNA. Okay, similarly there is one more enzyme, ribonuclease P. So ribonuclease P, ribonuclease phosphate, P means here. So these are the two exceptional cases of enzymes which are made up of nucleic acids. So they are made up of nucleic acid, but all enzymes are made up of proteins. But there is an exceptional case where nucleic acids they are made up of, means exceptional case where these enzymes are made up of nucleic acids, that is RNA, and as well means with made up of RNA, that is ribozyme, and another one is ribonuclease P. So let us see something about these enzymes. So if you see the another one that is called as a Holo enzyme. So let us discuss Holo enzyme. So it has two parts. So let us see what is a Holo enzyme first. So Holo enzyme. So it has two parts. So it has two parts in that particular enzyme. It is composed of two parts actually. So what are the parts means? So it has the protein part and as well as the protein part and non-protein part. It has a protein part of the enzyme and as well as non-protein part of the enzyme. So such type of enzyme is called as a holoenzyme, so non-protein part. So enzyme having two parts, the protein part and as well as non-protein part. Such type of enzyme is called as a holoenzyme. So let us see in detail about this particular holoenzyme. So let us see, this is the most important thing. So holoenzyme, so let us see something about holoenzyme. So good evening students. So good evening Gamma. So welcome to Unacademy EAMCET YouTube channel. So we are discussing the topic, the name of the topic, so that is enzymes. We are discussing the topic in today's class. So this holoenzyme, if you see this holoenzyme, so holoenzyme is having, it is composed of two components. So let us see what are the two components of holoenzyme. So holoenzyme is having protein part. So the protein part of enzyme is called as a apoenzyme. So it is having apoenzyme and so it is having non-protein part. Non-protein part is called as a cofactor. So non-protein part is called as a cofactor. So non-protein part. So non-protein part is called as cofactor. So protein part of enzyme. So protein part of enzyme. So protein part of enzyme is called as a apoenzyme. So protein part of enzyme is called as a apoenzyme. Non-protein part of the enzyme is called as a cofactor. So whole enzyme is having two parts. So first one is the protein part. The protein part is called as apoenzyme. So non-protein part is called as a cofactor. So first, once again, if you so this apoenzyme, if you classify further this, so this apoenzyme, so anyhow, so this is the protein part of enzyme, that is apoenzyme. Let us continue with the next one, that is cofactor. So let us see what is this cofactor. So if you see cofactor, so this cofactor, so just we'll draw in another slide actually, there is some. We'll draw from the first students actually. Okay, so holoenzyme. So first we will see the holoenzyme. So here I will draw a protein part. Okay, so holoenzyme is having two components, that is apoenzyme and another one is called as a cofactor. Okay, same thing. So apoenzyme, so what is apoenzyme? So protein part. So protein part of enzyme. So protein part. So protein part of enzyme. So protein part of enzyme is known as apoenzyme. So protein part of enzyme is known as apoenzyme. So another one is, this is cofactor. So cofactor is a non-protein part of the enzyme. So non-protein part. So non-protein part of enzyme is called as a cofactor. Non-protein part of enzyme is called as a cofactor. Now this cofactor is further classified into two types. So this cofactor, so a cofactor can be of two types. It is organic cofactor and another one that is called as inorganic cofactor. So it is of two types again. So organic cofactor and inorganic cofactor. So it is of two types, organic cofactor and inorganic cofactor. This organic cofactor, so it is of further classified into two types. So this organic cofactor includes prosthetic group. So it includes prosthetic group, and this is called as coenzyme. Okay, this inorganic cofactor. So inorganic cofactor includes metalloenzymes. So this is how these enzymes are classified, students. So Holoenzyme. So let us see this Holoenzyme. So Holoenzyme has this Holoenzyme. This Holoenzyme is having two components. Holoenzyme is having two components. So Holoenzyme. So we have seen simple enzyme. Simple enzyme is having only single component. So that it is totally made up of protein. So that is called as a simple enzyme. Now we are seeing Holoenzyme has two components. Holoenzyme. So Holoenzyme is classified into two types. It is having two components. The protein part of the enzyme. So protein part of enzyme is called as apoenzyme. So the protein part of enzyme is called apoenzyme. So non-protein part of enzyme is called as a cofactor. So non-protein part of enzyme is called as a cofactor. So protein part of enzyme is called as apoenzyme. So non-protein part of enzyme is called as a cofactor. So non-protein part of the enzyme is called as a cofactor. Then again, so this cofactor is again classified into two further types. It is organic cofactor. So it is organic cofactor, and another one is called as inorganic cofactor. It is a further classified taken two types, that is organic cofactor as well as inorganic cofactor. So this organic cofactor means the cofactor which is an organic compound. It is of two types again, prosthetic group, and second one is called as a coenzymes. It is prosthetic group and coenzyme. So organic cofactors are two types, students. It is prosthetic group, and as well as second one is called as the prosthetic group, and second one is called as coenzyme. So if you see the best examples of this prosthetic group, so example, if you see the example of prosthetic group, so it is a heme. So heme is the best example of prosthetic group. Then we have another examples of coenzyme. Coenzyme examples, if you see NAD, NADH, FADH2. So these are all the examples of coenzymes. So metalloenzymes. So metalloenzymes like for example, like if you see some metalloenzymes, so best example of metalloenzymes like for example catalase. So this enzyme catalase. So catalase is having the iron. So like that. So organic cofactors. We were discussing the organic cofactors. So what are the different types of organic cofactors? So we have seen organic cofactors. So organic cofactors, there are of two types we are mentioning. So prosthetic group. So how do we define this prosthetic group? So that is the question. So prosthetic group. So you would just see back once again. So prosthetic group. So what is a prosthetic group? It is an organic cofactor. So how do we define prosthetic group? You see this. So it is an organic cofactor. So it is an organic cofactor. So it is an organic cofactor that is tightly bound. It is an organic cofactor tightly bound to the apoenzymes. It is tightly bound to apoenzymes. So that is the important point you have to remember. So prosthetic group, so it is an organic cofactor which is tightly bound into the apoenzyme. So this is the important point we have to remember. It is tightly bound into the enzyme. So prosthetic group, it is an organic cofactor which is tightly bound to enzyme. So best example is, just now we have seen, it is the heme. So heme is the best example. So prosthetic group, it is an organic cofactor that is tightly bound. So similarly another organic cofactor, you see coenzyme. So coenzyme is another one. So coenzyme. So if you see coenzyme, how do we define coenzyme? So it is also an organic cofactor. It is an organic cofactor. So it is an organic cofactor. So loosely bound. So it is an organic cofactor loosely bound to the apoenzyme. So organic cofactor which is loosely bound to apoenzyme. So apoenzyme is a protein part of enzyme. So like this we will classify organic cofactor. It is a so organic cofactors are two types. So first one is called as a prosthetic group, second one is called as a coenzyme. So how do we define this prosthetic group? Is it is an organic cofactor that is tightly bound, to apoenzyme, that is called as a prosthetic group. Best example is heme. So similarly, if you see coenzyme, so what is this coenzyme? It is also an organic cofactor. So it is a organic cofactor that is loosely bound. So that is loosely bound to the apoenzyme. It is not tightly bound, but it is loosely bound to the apoenzyme. So let us see some examples of this coenzyme. If you see examples of coenzyme, so NAD, so NADH. So if you see the full form of NADH, so nicotine amide adenine dinucleotide hydrogen. So nicotine amide adenine dinucleotide hydrogen, that is called as a NADH. Similarly FADH2. So flavin adenine dinucleotide. So flavin adenine dinucleotide hydrogen. So like that we discussed. Okay. So once again, just go back, students. So Holo enzyme. So Holo enzyme, simple enzyme we were discussing in previously. So what is simple enzyme? So particular enzyme. So that particular enzyme has only one type of protein. Such type of enzyme is called as a simple enzyme. Okay. So if the enzyme is having only one, the enzyme is having only one component and it is made up of protein, that is called a simple enzyme. So here in this case, the enzyme has two components. The enzyme is having two components. So enzyme is having the two components. It is called as a Holo enzyme. So what are the two components of that particular enzyme? If you see the two components of that particular enzyme, it is the cofactor and as well as, so two components are cofactor and as well as apoenzyme. So what is the cofactor? The non-protein part of the enzyme is called as a cofactor. The protein part of the enzyme is called as a apoenzyme. So Holo enzyme has two components, the protein part and non-protein part. The protein part of the enzyme is called as apoenzyme. The non-protein part of the Holo enzyme is called as a cofactor. And this cofactor is again further classified into two types, organic cofactor as well as inorganic cofactor. So this organic cofactor is further classified into prosthetic group and coenzymes. Inorganic cofactor is further classified into metalloenzymes. Best example is like catalase, peroxidase. This all comes under the metalloenzymes. So if you how to define this metalloenzymes or inorganic, if you see the metalloenzymes, so these are also inorganic cofactors which is loosely bound to the apoenzyme. So like that we can define this metalloenzymes also. So prosthetic group, if you see the prosthetic group, how do we define this prosthetic group means? So it is an organic cofactor that is tightly bound to apoenzyme. So coenzyme, it is an organic cofactor loosely bound to apoenzyme. So best example of prosthetic group is heme. The best example of the coenzyme is NADH and FADH2. Now let us see how the enzyme reduces the activation energy in this particular diagram. So before that, so how do these enzyme functions? If you see, so every enzyme, enzyme react with substrate. So enzyme react with substrate. So this is how we represent. So enzyme react with substrate. To form enzyme substrate complex. So enzyme react with substrate to form enzyme substrate complex. So this is after that enzyme reacts with enzyme substrate complex. Then further enzyme product. And this enzyme gets recycled. So enzyme will be going out. So enzyme will be involving in the recycling process. And there is a formation of the product takes place. So here it is a reversible reaction. So like this enzyme react with substrate and forms enzyme substrate complex. So enzyme substrate complex formation, it is a transient state. So it is the enzyme substrate complex formation. This is a transient state. So transient means it is a temporary state. It only stays for some time. So it is a temporary. Okay, only for very short time it stays, temporary state. So after that enzyme substrate complex further develops into enzyme product, and later what happens, enzyme gets recycled. So enzyme will be recycled. So like this, what happens is enzymes. So what is the main function of enzyme? Just now we're discussing, they act as a biocatalyst. Enzymes act as a biocatalyst. So enzymes act as a biocatalyst, and so they reduce the activation energy. So they reduce the activation energy. Reduce activation energy. So they reduce activation energy. So you can see in this particular diagram how they reduce the activation energy. If you see this diagram, so now let us see this diagram clearly, students. So this is the diagram showing that how the concept of activation energy. So now this is the x-axis shows how the reaction is progressed. On the y-axis we have energy, that is potential energy. Now at this location, so we'll be having the substrate. So substrate is there. So substrate is having some energy. You can see there is some amount of energy is there. So this substrate react with enzyme. So the substrate react with enzyme, and here you can see this line. So this pink line. So you can clearly see this pink line. So pink line. Okay. So here it forms enzyme substrate complex. So when there is a presence of enzyme, so enzyme substrate complex will be formed here. So if you remember clearly, so enzyme substrate complex has more energy. So be clear from this particular diagram, if you clearly observe, so which is having more potential energy. So more potential energy, if you see the potential energies, so if you compare enzyme substrate complex is having more potential energy. It is having more potential energy than compared to, so it has more potential energy than compared to, okay, it is having more potential energy than compared to substrate, then compared to the substrate and product. So substrate and product, you can see the, so product, you see here this is the product. Here the product formation took place. Here the energy, there is no energy at all. So now this type of reaction, this is an exothermic reaction actually. Now this is an exothermic reaction. So enzyme substrate complex, is having more potential energy. So that is the important point we have to remember in multiple choice question will ask you. When enzyme react with substrate and during the formation of the product, these are the sequential events. In these events, which is having more potential energy means enzyme substrate complex is having more potential energy. So due to presence of enzyme, so what is happening is activation energy. So the amount of activation energy, is used is very less. So if you see without enzyme, so this is without enzyme. So you can clearly mention it is. So without enzyme, what happens? So without enzyme, more activation energy is required. So more activation energy is required. Without enzyme, the reaction is of course completing and formation of product is taking place, but more activation energy is required. More activation energy is required in this case. More activation energy is required in this case. So whereas here, if you see here, so activation energy in this case, you see less amount of activation energy, low amount of activation energy, low amount of activation energy. So low amount of activation energy. So low amount of activation energy is because of the presence of the enzyme. Due to presence of enzyme, it is requiring low amount of activation energy. So like this, what is the main function? So enzymes main function is they act as a biocatalyst. So what is the full form of biocatalyst? Means they act as a biological catalyst. They are acting as a biological catalyst. And what is the main function? They speed up the reaction. For every 10 degrees centigrade rise, the rate of reaction doubles. Okay. So main function of these enzymes is it reduces the activation energy and speeds up the chemical reaction or biological reaction, whatever may be. So from this given diagram, you clearly see that the potential energy is higher for enzyme substrate complex than compared to the product and as well as the substrate. Because of that reason, this type of reaction is called as exothermic. So this type of reaction is exothermic. So exothermic reactions has enzyme potential energy more for enzyme substrate complex than compared to the product and as well as the substrate. So that is about the today's class about the enzyme. Students, see you in the next class with a few more topics of this particular enzyme. So before completing the session, so once again, so I would like to discuss about our Unacademy Plus subscription. So you can see here, Unacademy is providing the EAMCET Ranker batch is going on, students. If you are interested, you can join this EAMCET Rankers batch by using my code Rajendra Live. Similarly, if you are interested for Telugu batch, so you can use my code Rajendra Live and join this Telugu batch, students. So certainly it will be benefit for you. So, and there is a best long-term plans also available. So you can go for long-term plan, 6 months plan costing around 10,125 rupees and 12 months plan costing around 12,375 rupees. So these are the best plans. So join this plan, students. So thank you, students. See you in the next class.